It's not just human beings that are attacked by viruses, but bacteria too. However, bacteria have their own way of defending themselves. How? By cutting the virus genome up into small pieces and preventing it from reproducing. And they do so with targeted precision and accuracy. For this, they use a special resource known as the CRISPR-Cas genetic scissors. CRISPR-Cas is a versatile two-part system. The precision is ensured by RNA molecules that reliably recognize the target sequences on the DNA. The cleavage through the DNA strand is carried out by enzymes that belong to the group of Cas proteins. Both parts of the system work together seamlessly. The RNA molecule guides the Cas enzyme towards the target sequence. The Cas enzyme then cuts through the DNA at precisely the predetermined site. We as human beings have learned to use CRISPR-Cas for our own purposes. We have fashioned it into a tool that allows us to intervene in depth into the genome. To this end, the RNA molecule is replaced so that the Cas enzyme can be directed towards a new target. As a result, any vegetable, animal, or human gene can now be altered. Unlike viruses, human cells are able to survive a CRISPR-Cas intervention. Natural repair processes fuse the severed DNA strand back together. However, the DNA sequence also changes during this process. A number of genetic letters are lost, with new letters filling the gap. If a gene is affected, it then usually loses its function. In the case of certain hereditary diseases, this is beneficial. Switching off a defective gene can protect the body from further damage. CRISPR-Cas therefore offers the possibility of using somatic gene therapies to treat individual organs specifically. This can alleviate severe hereditary diseases such as sickle cell anemia. However, the use of CRISPR-Cas in humans is not without risk, as the system sometimes makes mistakes. Repairing the DNA strand can alter the target gene in ways that are undesirable. This is referred to as the on-target effect. In rare cases, the genetic scissors also sever the genetic material in places not intended for this purpose. In a worst-case scenario, this off-target effect might cause cancer. CRISPR-Cas also enables what's known as germline therapy, where the genetic scissors intervene in the genome of eggs, sperm, and early embryos. These changes are then passed on to all subsequent generations. Such an impact is not yet foreseeable, which is why in Germany and in many other countries, germline therapy is banned.